And from the previous things I've talked about, I'm gonna touch on or talk about the cloning cloning on how to, well, it's the four stages of genetic engineering, and the four steps are DNA cleavage, production of recombinant DNA, cloning, and then the cloning screening. And and to begin, we start off with stage one, which is up here. And here we have the restriction endonuclease. They cleave the source of DNA to fragments, and they do the same with the plasmid. And there's because the recognition sequence is likely to occur many times. There's going to be a lot of fragments created from this, and a difference that could be obtained could be a different set will be obtained by using the endonuclease that recognize different sequences and can be separated according to size by using electrophoresis. And after this first step is complete, they're just getting the fragments out right here. You got the gene of interest. Over here the plasmid begins to cleave also. And it's important to know that the same endonuclease is used, and it's important to know that because if the same one is used, they could both be combined together to sticky ends. And in the second stage, we have the production of recombinant DNA. Basically, the fragments are placed into plasmids or viral vectors. A vector is basically anything that takes the gene of interest into a cell, a bacterial. And here we have they're cleaved by the same restriction endonuclease, and the two sources are mixed together and they form together. That's basic. It's basically saying that they combine, they mix. And from this we move on to stage 3, which is the cloning stage. The plasmid or virus serve as vectors that introduce the DNA fragments into cells, or to the bacteria more specifically. So the vectors, they move them into the cell. And then from here, as the cell reproduces, it forms clones of the cells that contain the fragment. And each one is maintained separately, and all of them together constitute a clone library of the original source DNA. And after the plasmids are inserted, we move on to the final stage, which is to identify the fragments, which are going to be the genes of interest. And there's a long process for this. You see that we have a lot of clones here. There's a clone, it's called the clone library, and basically they have to go through immense research or touch upon which one is the gene of interest. And in order to do so, there are basically two ways to do this. And you initially they eliminate any clones that do not match or contain vectors, as well as the clones whose vectors do not contain fragments of the source DNA. The first category of clones can be eliminated by employing a vector with a gene that confers resistance to a specific antibiotic, such as tetracycline. And only those vectors that are resistant to it are going to be able to grow, and the rest will be eliminated. And that narrows down the search. And then you have to eliminate vectors that don't have an inserted DNA fragment. There's a vector. You have to use a vector that contains LAC-Z, and the antibiotic resistant gene, which is required to make beta galactosidase. It's an enzyme that enables a cell to metabolize the sugar X-gal, and metabolism of the X-gal results in the cells with functioning LAC-Z to turn blue. So if it's able to metabolize X-gal and it has a functioning LAC-Z, the cell will turn blue. And if the endonuclease whose recognition sequence lies on LAC-Z, the gene will be interrupted and the recombinants are formed, and the cell isn't going to be able to metabolize X-gal. Basically, if the recognition sequence is on LAC-Z, the gene's going to be interrupted and they won't be able to metabolize xCal. And cell with vectors that contain the source DNA are going to stay colorless. And so the ones that are going to be wanted here are blue. So here we have what I was talking about. The blue ones move on because those are the ones that actually work, and the colorless ones is because the DNA fragment is inserted within the LAC-Z gene, which isn't able to metabolize the xCal, and it remains colorless. The second way to locate the gene of interest is called hybridization, which is clone genes form base pairs with complementary sequences on another nucleic acid, and called the probe. And here we have the process, which is it's quite simple. You basically get the bacteria and you grow them on an agar, and from here you make a replica, which you by just pushing a filter against the colonies, and some of the cells are gonna stay on there. And from this, you wash the filter with a solution that denatures the DNA, and it contains radioactively labeled probe, and the probe contains complementary sequences to the gene of interest and binds to them. And this is visible after you remove it, and those that contain the gene are gonna keep the probe and gonna show radioactivity on the film. And you could identify it by, well, just whatever is radioactive. You get to pinpoint it on the agar and find out which colony had the gene of interest. And you just, at the end, just compare them and find out which one had it. And this is basically how to find everything I talked about was how